Hello everyone and welcome to Pocket Size Bio with Dr. Casco. In today's video, we're going to talk about Chapter 10, Photosynthesis of the Campbell Biology textbook. We're going to look into how photosynthesis feeds the biosphere, how it converts light energy to chemical energy of food, how these light reactions convert solar energy to the chemical energy of ATP and NADPH, different from cellular respiration, and how the carbon cycle uses the chemical energy produced in the light reactions to reduce carbon dioxide to sugar. Aside from that, we're going to talk about alternative mechanisms of carbon fixation and how they have evolved in hot, arid climates, and then finish off with a little bit of review on photosynthesis. So let's get to it. Okay, section 10.1 is photosynthesis feeds the biosphere. Now, this section isn't anything new. We've seen this since elementary school, science, but it's always good to review on the key concepts, right? Life on Earth is solar powered. Chloroplasts use photosynthesis to capture light energy. Ultimately, that light energy will be converted or transformed into chemical energy stored in sugars and other organic molecules. Okay, photosynthesis nourishes almost all of the living world, directly or indirectly. There are some organisms that are independent of photosynthesis that you can find them in hydrothermal vents deep in the ocean floor. Okay. So organisms obtain organic compounds by autotrophic nutrition. We'll talk a little bit about that in a little bit. Or heterotrophic nutrition. Okay. And autotrophs, okay, these organisms that are referred to as autotrophs, they produce organic molecules from carbon dioxide and other inorganic raw materials. All right. And ultimately, they're the source of organic compounds for all heterotrophs. They are, in a common sense, producers of the biosphere, as we know them commonly, right? Almost all plants are autotrophs. They require water, some minerals, carbon dioxide. More specifically, even from the word autotroph, plants are photoautotrophs. So they use light to synthesize organic compounds. Okay. Photosynthesis also occurs in other organisms like algae. You have some photosynthetic protists, some prokaryotes can photosynthesize. Also, there are some variety of life there in terms of autotrophic organisms. You have some heterotrophs now. These are organisms that live on organic compounds that are produced by other organisms by the autotrophs. Right, so they are the consumers of the biosphere. Among these, you have the animals, right? And others are decomposers that feed on dead organisms, organic litter. Okay, then they're mostly fungi. Some prokaryotes are able to obtain nutrients in this way. Okay, and you can see some examples here in these figures here, figure 10.2. Photoautotrophs, right? You have your plants, you have multicellular algae, okay, unicellular protists, cyanobacteria, even purple sulfur bacteria. In this particular case, we're going to see in a little bit on these cases here. Ultimately, you have these organisms that are able to, process these organic compounds, die out a long time ago, and they produce fossil fuels. Right? They're formed from the remains of organisms that died hundreds of millions of years ago. Okay? Ultimately harnessing that sun's energy. Okay? Fossil fuels represent stores of the sun's energy from a long time ago. Okay? From distant past. Because fossil fuels are not renewables, of course, researchers are exploring photosynthesis for alternative fuels. Okay? I recently read an article of how a research lab was able to create a synthetic photosynthesis or artificial photosynthesis process, and is able not just to produce organic compounds, but actually precursors for drug molecules. And I might make a video of that later on. Okay. All right. Going to the next section. Going to the next section here. 10.2, photosynthesis converts light energy to the chemical energy of food. All right, so photosynthetic enzymes and molecules, they're grouped in a biological membrane for efficiency. We're going to see this very similar to cellular respiration and the electron transport chain kind of situation there. And photosynthesis likely originated in bacteria that had infolds of the plasma membrane. So it kind of infolded in particular regions, creating these double membranes. Okay, and existing photosynthetic bacteria, those infolds, okay, function similarly to chloroplasts' internal membranes. Yeah, so that's again, if you remember that endosymbiotic theory, that endosymbiont theory, suggests that the original chloroplast was actually a photosynthetic prokaryote living inside an eukaryotic cell. Okay, just a little bit of a refresher there from the previous chapters. Focusing now on chloroplasts. Chloroplasts are the sites of photosynthesis in plants. All green parts of a plant have chloroplasts. Okay, particularly the leaves, as we all observe in our daily life there. They are the major sites of photosynthesis. Okay, approximately 500,000 chloroplasts per millimeter square of leaf surface, contains all these organelles, right, of chloroplasts. And if you were to zoom into the leaves, right, the leaves have particular sections and particular tissues that you do need to know and familiarize yourself with. If you take a look at this diagram here, you have a leaf. If you look at the this little kind of cross section here of the leaf, you see there's different layers here. You have a mesophyll layer, and there's two types of mesophyll layer. This is essentially where the photosynthetic cells are that contain the chloroplast. And in particular, so you have these mesophyll layers that house a lot of chloroplasts, okay, that interior leaf tissue. Okay. Aside from that, within the leaf, generally on the underside, you have these specialized structures, again, specialized cells. They're called stomata. We're going to talk about these in a lot more detail in the later sections of this chapter. And oxygen and gas exchange occur at these sites, right? Oxygen exits, carbon dioxide enters through the stomata, and they're little pores that you can find on the leaf. Okay. Aside from that, plants do have vascular systems similar to animals, right? They have veins where you can deliver water from roots and carry sugar to non-photosynthetic areas. You have your xylems and phloems that we'll see later on. Okay. Focusing a little bit on the mesophyll now, a mesophyll cell can have 30 to 40 chloroplasts, okay, which have a size about 2 to 4 micrometers, right, in diameter. Okay, and this can vary, right? And each chloroplast itself has two membranes, as we know already from the tour of the cell chapter. And as you see here, within the inner membrane, you have a dense fluid called the stroma. Okay, so taking a look at this particular diagram as I go along, when you focus on the stroma, there's some more structures suspended in the stroma. You have these flattened sacs. They're called thylakoids. And here's where the magic happens of photosynthesis, right? It's an internal membrane system. Just going to zoom in to these little diagrams here. So you have your outer membrane, you have the inner membrane. In between these two, of course, you have the intermembrane space. And then within the inner membrane, you have the fluid stroma. And then it houses another kind of structure, which is the thylakoid. So these little kind of flattened pancakes, or these little sacs, can be stacked together even. And when they're stacked together, they're referred to as grana. Okay, or granum, one stack. Okay. Very important, the thylakoid space to mention, right? So even within these little thylakoids, you have a fluid space here. That's called the thylakoid space. And that's going to be very important when we talk about the electron transport chain. Okay? So, again, they have that thylakoid space. Very, very important for you to remember these structures. Okay? And then, further on within the thylakoids, you have chlorophyll. Chlorophyll are pigment molecules. They're green pigment molecules that are located in the thylakoid membranes, right? And chlorophyll plays an important role in absorbing light, different wavelengths of light, okay? Ultimately absorbing that energy to promote photosynthesis. Photosynthetic membranes of prokaryotes arise from infolded plasma membranes, okay, which are also called thylakoid membranes. So it's interesting to note there. All right, going into a little bit more now in terms of the chemistry of photosynthesis and the chemical equation. So tracking atoms through photosynthesis. So essentially, how do we know we know the reactants, the atoms like the carbon and carbon dioxide and the oxygen from water, where does it end up within the products? So when you have this reaction being powered by light, photosynthesis produces organic compounds from organic compounds and oxygen, okay, from carbon dioxide and water. The equation you can see here, right? You have 6 molecules of carbon dioxide, 12 molecules of water, plus light energy, all working together to produce glucose, 1 molecule of glucose, plus 6 molecules of oxygen, and 6 molecules of water. Okay? So, in this reactions, or this series of reactions of photosynthesis, you don't directly produce a glucose molecule. You actually produce a 3-carbon sugar, which is G3P. We're going to see this later on. Water does appear on both sides. So if you kind of simplify the equation, you have 6 molecules of carbon dioxide and also of water, plus light energy yields 1 molecule of sugar and 6 molecules of oxygen. Okay? What we already know from high school, right? So this overall chemical change and chemical reaction is the reverse of cellular respiration. These both processes occur in plants, so definitely keep that in mind. Okay, cell respiration and photosynthesis occur in plants, but of course, chloroplasts do not simply reverse the respiration steps. It's a very complex process. Okay. If you were to break down this equation to its simplest form, it would look something like this, right? 1 carbon dioxide molecule plus 1 water molecule yields this organic compound here, a carbohydrate, that base equation of a carbohydrate, sugar, and oxygen. Okay? And scientists wanted to know how this actually worked and how they passed where the atoms go, right, within the reactants and the products. So the splitting of water, right, scientific inquiry here. So evidence that chloroplasts split water molecules, okay, enabled researchers to track atoms through photosynthesis. And one clue that scientists noted, right, is that oxygen O2 molecules given off by plants comes from water and not carbon dioxide. So you might think, being gas and gas, that carbon dioxide would ultimately change to oxygen. Okay, and this was the situation, right? Before the 1930s, the hypothesis was that photosynthesis split carbon dioxide. Carbon dioxide would break down into carbon and oxygen gas, and then carbon would react with water to produce, this sugar compound. Okay, you have a scientist called Van Niel challenged this hypothesis using bacteria that use hydrogen sulfide instead of water. Okay, so you're using sulfur bacteria. Okay, so these bacteria produce sulfur as a waste and not oxygen. Okay, so he proposed for these special bacteria that if you have carbon dioxide and you have hydrogen sulfide, this would yield, your sugar base, water, and 2 atoms of sulfur. So he generalized for plants. So carbon dioxide plus water would yield this particular equation here. So he hypothesized that if this is what's happening for plants, can something similar be happening with this particular reaction in sulfur? So he hypothesized that the plants split water as a source of electrons. Okay, as a source of electrons. So keep that in mind. Water as a source of electrons from hydrogen atoms releasing O2. And if that's the case, you would have something like this. So carbon dioxide, you would have hydrogen sulfide, would yield a sugar compound plus water plus, your X molecule. So 20 years later, scientists confirmed Van Niel's hypothesis, using oxygen 18 as a tracer. So essentially, any compound that provides the electrons and the hydrogens can produce water, and you'll have that exit molecule, if I can say, like sulfur. And in this case, you have oxygen. Okay. So water, or this hydrogen sulfide, can serve as donors of electrons. Okay. So scientists later confirmed they labeled carbon dioxide, or they labeled the oxygen in water. Okay. And then what happened? The oxygen 18 appeared in the O2 only when water was the source. Okay, only when water was the source. So hydrogen from water is incorporated into the sugar, okay, and oxygen is released. So ultimately, you get this kind of scenario where you have sulfur bacteria. They're using hydrogen sulfide, okay, react with carbon dioxide through a series of reactions. Ultimately, you get your sugar, you get water, and then you get your, whatever X molecule is in that beginning reactant. Okay. Good. So ultimately forming this here. So tracing atoms through photosynthesis, the atoms from carbon dioxide are shown in magenta, and the atoms from water are shown in blue. Okay. So the reactants, the carbon goes, of course, into the sugar. Okay, the oxygen from carbon dioxide goes also into the sugar and also into water. But the oxygen coming from water, okay, ultimately is liberated as gas, oxygen gas. Okay, so it's the water. The whole point of the experiment is that water is what yields into this this oxygen gas molecule. okay, as opposed to carbon dioxide. Carbon dioxide is used to produce water and sugar. Okay, let's go to the next part here. So photosynthesis is a redox reaction or a redox process. And similarly to what we've seen in aerobic respiration, okay, similar things occur in photosynthesis. In terms of cell respiration, energy is released as electrons fall toward oxygen. If you remember the electron transport chain, and electrons are passing from one acceptor and one donor and so forth and so forth, one compound gets reduced, the other one gets oxidized, and so forth and so forth. A similar procedure happens here in photosynthesis. Okay. Photosynthesis reverses the direction of electron flow. Okay. And we're going to see this a lot more detail when we get into the actual ETC here. But ultimately, water is split. The splitting of water, the breakdown of this water, will liberate electrons and hydrogen plus ions, right, that are transferred from water ultimately to carbon dioxide, reducing. So having this flow of electrons and having some hydrogen plus ions will be used to reduce sugar or to produce sugar. Okay, so electrons increase in potential energy, ultimately are moving from water to sugar, and that will require some source of energy to promote this whole process, which is light. Okay, so just giving a preview of what's to come in this process of photosynthesis, we have two stages. Light reactions. They're also referred to as light-dependent reactions. And then you have light-independent reactions, which is ultimately the Calvin cycle, otherwise known as the Calvin cycle. In light reactions, you have a conversion of solar energy or light energy to chemical energy. Okay? And this is what you really need to know, the inputs and the outputs of each of these reactions. In the light reactions, water is split, and it will provide a source of electrons, hydrogen plus ions, and will liberate oxygen gas, O2. Okay. In the going into detail now, the light reactions, they absorb light via chlorophyll, and the chlorophyll will ultimately pass down the electrons and jump from molecule to molecule. They'll harness the light energy to excite electrons, and then these electrons will start passing down from molecule to molecule, complex to complex. Okay. Ultimately, you have electrons and hydrogen plus ions to produce NADPH. So NADPH in this case is the last electron acceptor. You have its oxidized form, which is NADP plus. So this is something you definitely need to remember. I'm going to highlight that, which will form NADPH. Okay, and you also produce ATP via chemiosmosis. If you guys remember that use of hydrogen plus ions. Okay? This is also known as photophosphorylation because it's using light. All right. So light energy ultimately is converted to chemical energy in NADPH, having that reducing power NATP. So make sure you really get that down there. Okay, there's no sugar being produced yet. This happens in the Calvin cycle. This is the light independent reactions or the Calvin cycle. The Calvin cycle is named for Melvin Calvin, who worked out its steps, who figured out these steps in the 1940s. Interestingly, right? The cycle begins with carbon fixation. Again, we're going to see each of these stages in a lot more detail, just to give you a brief summary of what's to come. There's an incorporation of carbon dioxide into organic molecules, so it's very similar to cell respiration, right? Where you are getting acetyl coenzyme A coming into a series of molecules. Here, you're getting a similar thing. Carbon dioxide will come in and attach to molecules and then go through a cycle and process the carbon in the carbon dioxide. Okay, fixed carbon is reduced with electrons from NADPH, so we're going to take a look, and ATP from light reactions also powers certain parts of the Calvin cycle. Ultimately, at the end of this cycle, one of the can't say the end of the cycle, as the cycle goes through, you make a 3-carbon molecule. Okay, and when you make 2 of these 3 carbon molecules, you ultimately produce sugar. Okay, as I mentioned before, this is a light independent reaction because it doesn't really require light directly. Sorry about that. Okay, so the Calvin cycle in most plants occurs during daylight. So of course, during daytime we have the sunshine and the plants are absorbing light to, carry out the light reactions, and you're getting a steady supply of ATP and NADPH, ultimately occurring in the stroma for the Calvin cycle. Light reactions, of course, they need that ETC to be in a membrane. That membrane is the thylakoids. We're going to see that in a bit. Okay. Ultimately, in the thylakoids, you have NADP and ADP. They're coming in. These are the oxidized forms. We need to give them some electrons. We need to give them some phosphates. Okay, ATP synthase is found here also, and so forth. Ultimately, to produce NADPH and ATP. Okay, and this is released in the stroma for the Calvin cycle. This is a visual representation of what we're going to look at. Water comes in with light. Also, you have NADP plus and NADP plus inorganic phosphate. All of that comes together in the light reactions to produce these main things, ATP and NADPH. Oxygen goes out of the plant into the air, into our lungs, right? And then you have carbon dioxide entering in the Calvin cycle in the stroma, using the products of the light reactions, ultimately to produce a sugar. Okay, let's continue on. We do need to talk about light in itself. So, section 10.3, the light reactions convert solar energy to the chemical energy of ATP and NADPH. A little bit of physical science here, just to review. So light in itself is a form of electromagnetic energy, okay, radiation, and it travels in rhythmic waves. And being an electromagnetic wave, these are disturbances of electric and magnetic fields that are passing, from the source to whatever it hits. Travels through space in a vacuum. It doesn't necessarily have to travel through a medium. Okay, so you have these electromagnetic waves. In particular, as you know, the basic structure of a wave, you have crests and you have these troves and you have a wavelength. The distance between crests is the wavelength. Okay, and you can have different variations of wavelengths in terms of how long they are. Right? They can range from very, very small nanometers. And this is what you have here, the electromagnetic spectrum. Okay, let me just zoom in a little bit, where you have gamma rays being the shortest wavelengths, and then you have the longest wavelengths being radio waves that could reach kilometers of size in their wavelength. Okay, so this kind of spectrum of lengths of wavelengths is called the electromagnetic spectrum. Now, visible light is found in a very narrow range among this spectrum, okay, from 380 nanometers of wavelength. So you can see here, this wavelength of light, okay, where you get the purple, okay, color of light is the shorter wavelength. And then as you go and increase the distance, you're getting longer wavelengths and longer wavelengths till you get to this 750, 740 area. And this is basically the red wavelength. Okay, so here you're shorter wavelength, and as you go along, the wavelength is increasing. And as the wavelength increases, you're getting a different color of light. Okay, At the same time, shorter wavelengths carry higher amounts of energy. Okay? And as you go along, that energy decreases. Now, this is the visible light spectrum where we can capture with our eyes as humans, right? We detect that as color. Very, very important. So, just to note a little bit more, so light travels as a wave, but has properties of discrete particles known as photons. And photons, these particles of light, have fixed quantities of energy. Okay? Fixed quantities of energy. And energy, again, as I mentioned already, is inversely related to the wavelength. As wavelength increases, energy decreases. Wavelength decreases, energy increases. All right, so you have purple wavelengths versus red wavelengths, and they each have their own amount of energy. Okay, so the atmosphere, having gases floating around, filters most wavelengths of light. Visible light passes in significant quantities, and the light that hits plants and that hits, all the natural world, all these autotrophs, is what drives photosynthesis. Now, getting into the actual pigments within chloroplast, you have the photosynthetic pigments, the light receptors, right? So when light hits or meets matter, it could be reflected, transmitted, or absorbed, right? And specific molecules that are able to absorb visible light are referred to as pigments. Okay, that's their function, these substances. And different pigments can absorb different wavelengths of light. Okay, and that wavelength ultimately disappears and gets absorbed within these pigments. The energy gets absorbed within these pigments. Okay, and a leaf looks green, as we all know. We see them as a green, having a green coloration, and it's because chlorophyll absorbs the majority of colors, particularly in the red, spectrums of light and the violet blue colors of light, but it reflects greens, as you see here. Light comes in, hits these chlorophyll pigments. All the colors are essentially absorbed. I don't want to say all the colors yet. We're going to see the graph here. And green, the wavelength of green light gets reflected or transmitted passing through. Okay? But it doesn't get used really. And that is what we see in our eyes. Okay? So, yes, it reflects or transmits green. There are machines that can measure a certain pigment or molecule's ability to absorb light. They're referred to as spectrophotometers. Okay, spectrophotometers. Very, very important tool, right? To measure a pigment's ability to absorb various wavelengths. So, what happens here? You beam, narrow wavelengths through a pigment solution. Okay, as you see here in particular. Okay, you can diffract these different colors of light and see how a certain solution, a certain pigment, can absorb the that particular color, and then you can figure out an absorption spectrum to plot out, in this particular case of chlorophyll, light absorption versus that particular wavelength. Okay, so with that, scientists can figure out, what colors chlorophyll and other pigment molecules can absorb, what wavelengths they can absorb. Okay. Continuing on here. So this is what I was referring to as the this kind of absorption spectrum. So in plants, in chloroplasts and the thylakoids, there's several pigments that are present. You have chlorophyll A, and this is like the go-to one. This is the one you have in a good amount, right? It participates directly in light reactions. It absorbs best red and violet blue. and it absorbs the least wavelength, which is green. Okay, so if you take a look here, you have the wavelengths of light, and here on the y-axis you have the absorption of light by the pigments. So let's say chlorophyll A, you have this kind of dark turquoise, okay, line here. You see that it's absorbing a good amount in the purple, a little bit of blue here in the wavelength, and then green, not so much, and even yellow, orange, not so much also, until you get into the red spectrum here. Okay. But interestingly, plants have other types of pigments like chlorophyll B and carotenoids, which help in the absorption. Okay, so what I was mentioning again, you have this absorption spectra. So you have chlorophyll B. If you notice, there's a lot more blue being absorbed for chlorophyll B, and they absorb around again orange and a little bit of yellow here. And then you have your carotenoids, which are absorbing a lot of purple blue wavelengths. Okay, so this improves the efficiency at which plants can absorb different colors of light. Okay, ultimately harnessing more light energy, and so forth. Okay. You also have another graph, which is called the action spectrum. So this profiles the relative effectiveness of different wavelengths that are driving photosynthesis, okay, with these autotrophic organisms. Okay, so this measures changes in photosynthetic activity, basically oxygen release, okay, as wavelength varies. And I'm gonna go down a little bit just to show you the picture here, but here's the action spectrum. Okay, so Engelmann, a scientist that demonstrated this action spectrum, used filamentous algae and aerobic bacteria to measure oxygen production, right? So his results match modern day action spectra experiments on the efficacy on how you had these wavelengths being absorbed and the amount of products being made. Okay, and you see how here there's a lot more aerobic bacteria being present on these particular wavelengths. Okay. Sorry about that. Okay. As we get into the different, kind of pigments, but we focus on the ETC, an electron transport chain, only chlorophyll A participates directly in the light reactions. And we're going to see that more in detail as we go along. So these accessory pigments, carotenoids and these chlorophyll Bs, they're referred to as accessory pigments, like helper pigments. Okay, they also help absorb light, but ultimately they're going to transfer that energy to chlorophyll A, and chlorophyll A will be the one that will drive the ETC. Okay, chlorophyll B has a slightly different structure, an absorption spectrum, and again, as I mentioned before, funnels energy to chlorophyll A. Okay. Talking a little bit more about carotenoids, you have hydrocarbons that they can manipulate various shades of yellow and orange, absorb various shades of yellow and orange. Okay, they could absorb a good amount of violet and blue-green light. Okay, again, ultimately they funnel energy from other wavelengths and participate also in photoprotection against too much light. If you have too much light, that could be a problem. This could damage the structure, right, of the autotroph. So by absorbing this, you can funnel energy well, and it can dissipate excessive light energy and ultimately preventing damage from reactive oxidative molecules. Okay, carrots, they help in night vision, and they contain a lot of phytochemicals and these carotenoids like beta-carotene. Okay, and they're healthy food products. Again, with that same idea of preventing damage. Okay. This is a structure of chlorophyll A that you can see, not just chlorophyll A, but just chlorophyll in itself, and some differences between chlorophyll A and chlorophyll B is a functional group, right? As CH3 and COH. Okay. You have this particular structure called a porphyrin ring, and this is a light-absorbing head of the molecule, and there's a magnesium atom at the center. Okay, so we're continuing now with excitation of chlorophyll by light. So when a molecule absorbs a photon, an electron is elevated to an orbital with more potential energy. Generally, electrons are in their usual electron orbital, and you refer to that as their ground state, and they move from a ground state to an excited state. So what happens here is that electrons absorb energy and they can move to a different type, a different orbital. Okay, so a molecule can absorb only photons whose energy matches the energy difference between grounded and excited states. Okay, can only absorb photons. So if you have a photon coming in, electromagnetic radiation, and just to give you this little picture here, you have a grounded state of a molecule, chlorophyll here, and you have an electron in this particular energy. Okay, this amount of quantification from here to here. If you have a photon that has that same amount of energy, then you would get that electron being excited and moving to that excited state. Okay, so it has to match. Okay. This energy difference does vary among different atoms and molecules and for each particular pigment. So you have a unique absorption spectrum. Okay. And it's quite interesting when you energize these electrons, they have a higher amount of energy, and so they liberate that energy as heat, as fluorescence. Okay. So as heat as fluorescence, and if you were to isolate chlorophyll molecules in a beaker, right, or in this particular example here, you have a solution having these molecules in an Erlenmeyer flask, and by themselves, when you excite them, when you beam some light on these molecules, they will liberate energy via heat and fluorescence. So they'll illuminate, right? They'll fluoresce, they'll glow. Okay, and that in itself is what is called fluorescence. Okay, so they emit light. Isolated chlorophyll fluoresces in a red-orange coloration, right? Part of the spectrum then gives out heat. Okay, now of course plants don't do that. We don't see plants glowing like this. Okay, but so when you have chlorophyll that is excited by light, different things happen when they're part of a system within the chloroplast. Okay, so in the thylakoid membranes, chlorophyll is organized with, in complexes with proteins and small molecules to create what are called photosystems, and that's what we're going to see next here in this figure you see here. So a photosystem, okay, it's a complex of different molecules, and amongst this that you see here, you have different areas. You have a reaction center complex, and you also have a light harvesting complex. So let's take a look at what this is about. Okay, so in a photosystem, it's this whole general complex that you have here that is embedded within the membrane of the thylakoids. Okay, so make sure you memorize that. Where do you find this? They're in the thylakoids of the chloroplast. Okay, so on this membrane, you have a particular area that's called the reaction center complex. And if we take a look at this diagram here, you have this central portion here. And in this reaction center complex, you have what is called a primary electron acceptor, and you have this chlorophyll A molecule. Okay, so they're organized in a particular way where the electrons from a chlorophyll A molecule will jump to this primary electron acceptor. Okay, and I'll get back into that in a bit. And then you have your light harvesting complex, which is these surrounding complexes that are surrounding the reaction center, and they contain pigment molecules. And these pigment molecules could be chlorophyll A, chlorophyll B, carotenoids that are bound to proteins, all with that function to absorb and harvest a large a good amount of the spectrum of light. Okay, you also have a larger surface area. Of course, more surface area, more light being harvested, more energy being collected for the reaction centers. So the light harvesting complexes can act as like antenna for the reaction complex. Okay, so when a pigment absorbs a photon, energy is transferred to other pigments until it's passed down, it's relayed from one pigment to another, to another, to another. And there's a transfer of energy, as you see here. Energy is passed down until you have chlorophyll A molecules obtaining that energy, exciting electrons, and passing it to the primary electron acceptor. Okay. So at the reaction center, you have this primary electron acceptor that will gain that electron from chlorophyll A via redox reactions. Okay, via redox reactions. Again, that same idea that isolated chlorophyll fluoresces because there is no electron acceptor. Because there's an electron acceptor here, you don't see that fluorescence. It's just a relay. It continues on. The electron will shift to this next part that we're going to take a look at. Okay, so the electron's potential energy, okay, that should be dissipated as light and heat doesn't happen here because it's going to be passed down to another molecule, as we're going to see. Again, this is in the thylakoid. You have the thylakoid space, the interior of the thylakoid, and you have the stroma in the outside. Let's move on to the next panel here. Okay, so now that you understand what a photosystem is, there's actually two types of photosystems. Okay, and they're nearby each other. You have photosystem 2, designated as PS2, and photosystem 1, or PS1. Okay, and they each have their own characteristic reaction centers. There's some slight differences, the light reactions actually begins with photosystem 2. Okay, the reason why it's called photosystem 2 is just because that it was the first one being photosystem 1 was the first one that was discovered, and photosystem 2 was the second one being discovered. But in terms of function and relay of light harvesting and light reactions, photosystem 2 goes first. Okay, hopefully that's not tricky there. Just know that this starts with photosystem 2. Okay, and photosystem 2 has a particular chlorophyll A. Is designated as P680, because it absorbs, its peak absorption is 680 nanometers of wavelength of light, of visible light. Okay, and PS1 has a reaction center with a chlorophyll P700 that will maximize 700 nanometers of wavelength. Okay, so there's nearly identical chlorophyll A molecules that enable the absorption of these particular wavelengths of light, and they're pretty much side by side with the exception of the electron transport chain in between them. Okay, and they look something like this. So here you have a photosystem, and here you have a photosystem. Okay. So this is a photosystem in itself. Sorry about that. So the structure of a photosystem, and this is a computer model based on X-ray crystallography, if I'm not mistaken. Oh, no, my apologies. Yeah, it shows two photosystem complexes side by side. Okay. Continuing on, so now you have these two photosystems working together, ultimately to generate ATP and NADPH. Okay, don't get confused with NADH2 of cellular respiration. So here you have NADPH. And there's two ways in which you can harvest these things, right? ATP or NADPH. There's two routes. you can have a linear route, okay, or you can have a cyclical route here. And we're going to take a look at this linear route where the electrons flow in a linear manner. Okay, so here, what happens, starting off with photosystem 2, you see here, and I'm going to zoom in just a little bit as you can see better the diagram. Okay, photosystem 2 absorbs photons, so light, from the environment, and these harvesting molecules, pigment molecules, would absorb that light, and as they're passing from one to another to another to another, that electron is gonna get excited, right, from your P680 chlorophyll A. Okay, the electron is captured. As the electron gets excited, the electron gets captured, okay, by the primary electron acceptor. This electron in itself gets passed down, and then it'll go onto the electron transport chain. Okay, P680 gets oxidized to P680 plus. Okay, of course, because you're removing that electron. Now, what happens here, if you see on step number 3, okay, here's where the water molecule comes into play of the process of photosynthesis. An enzyme comes in and extracts electrons from water. Okay, so water is essentially split. It's broken apart into 2 hydrogen plus ions, which is released into the thylakoid inner space, and 1 oxygen atom is also liberated. And that immediately combines to form oxygen 2. So if you see in this little diagram here, okay, water is split into electrons. These electrons are given to this chlorophyll A molecule. Hydrogen plus ions are released. 2 of them are released, and oxygen is also liberated, quickly reacting with another oxygen molecule to produce oxygen gas. Okay, so you always have a reestablishment of electrons. Electrons don't get lost there. Okay, so just to zoom back, we can see the steps here. So these photo-excited electrons will then pass from the primary electron acceptor to an electron transport chain. Okay, slightly different from cellular respiration. You have your initial complex here, which is called plastoquinone. And then you undergo a series of cytochrome complexes, okay, as we pass the electrons down. And then you have your last one in the chain, which is plastocyanin. Again, as electrons are passing down, remember that whole function of the electron transport chain in cell respiration, ATP is being generated because you're having hydrogen plus ions being shuttled from one side to another to produce a electron, a hydrogen plus ion, a chemical gradient. Okay, so again, electrons are still passing down from one to another, and then they go to this second photosystem, photosystem 1, okay? And the reason for this is because as electrons are passing down from one to another, they're actually losing energy, okay? They're falling to lower energy levels because this energy is being used to harness ATP. Remember, hydrogen plus ions are being shuttled. They're passing down into the thylakoid lumen, the inner space of the thylakoid. And again, it's producing an electron gradient. Okay, so then what happens as electrons are passing to the photosystem 1, the chlorophyll molecules there are also harnessing light energy, and it's exciting an electron from photosystem 1's chlorophyll, which is P700. Okay, P700. So you have an electron here, and it's being shuttled out just because there's so much energy being absorbed by light. This goes to another primary electron acceptor, which will continue another electron transport chain. Okay, but what you need to know then is that electron that is lost, just to zoom out so you can see the picture again, okay, that electron that is lost here in P700 will be regained from the incoming electron of photosystem 2. So it's like a relay. This one leaves, and then another one comes in to reestablish it inside P700. Okay, so that hole, this is what I'm referring to as this hole, this is quite bare bones there, is filled by an electron from PS2 transport chain. Okay, and then again, this photoexcited electron is passed from that primary electron acceptor to a second electron transport chain via ferredoxin. So you have this particular molecule here, and then these electrons are passed down to NADP plus. Okay, remember this is an oxidized state of NADPH, and it's floating around with the help of an enzyme called NADP reductase. Okay, this will catalyze the electron to come in initially to ferredoxin and then to NADP plus, and then you get this reduced state of NADPH. So now you have an electron carrier bound to an electron, and this NADPH will go to the Calvin cycle, the next step of the of photosynthesis. Okay, so light reactions ultimately use solar power from photosystem 2 and 1 to provide ATP and NADPH in the Calvin cycle. Okay, and in this particular instance, of course, you have an electron flow. Okay, electrons come from here, then they go down the electron transport chain, and then they'll be relayed back, relayed to NADP plus to reduce to NADPH. So there's a net movement, okay, of electron flow. So this is what we're referring to as linear flow. Okay, this is a very nice analogy that you see here, where you have photons, basically light, providing that initial energy to jumpstart electrons to go into these electron transport chains. As the electron transport chain carries these electrons from one molecule to another, to another, to another, they generate ATP via hydrogen plus ions, okay? To re-excite or to add more energy to these electrons, more energy from light is captured, so electrons can be shuttled into higher energy states, okay, which ultimately provide NADPH with a good electron or that has good amount of energy. Okay, let's continue on. All right, then you have your cyclical flow of electrons. Okay, so under certain conditions, cyclic electron flow occurs, and this is only PS1, so only in the first electron chain. And I'm actually going to highlight that it occurs in the photosystem 1 rather than photosystem 2. Okay, this does not occur in photosystem 2, only photosystem 1. So, in our story here, where electrons are passing down, let's say electrons go inside photosystem 1 already. And what happens here is that the P700 chlorophyll A excites the electron and it goes to the primary acceptor. But instead of ferredoxin moving it to NADPH, ferredoxin will shuttle it back to the cytochrome complex. Okay? So electrons cycle back from ferredoxin to the cytochrome complex to continue going into the photosystem 1 and photosystem 1 and photosystem 1. So why do you think that occurs? It's ultimately to generate ATP. Okay? To generate ATP. No oxygen is being produced because you're not using, remember the oxygen is being split with water or from water in photosystem 2. Since you're not going back to photosystem 2, then that doesn't happen. So no oxygen is released and no NADPH is produced. You're only producing ATP. And this occurs in some photosynthetic bacteria, purple sulfur bacteria, okay? And in these particular mechanisms, you have this sole ATP generating mechanism of photosynthesis, okay? Evolutionary biologists hypothesize that these groups are descendants of bacteria in which photosynthesis first evolved. So it's kind of a more, if I can say, primitive flow or form or complex or pathway. Okay. Cyclic flow occurs in species with both systems. So some cyanobacteria and plants, they could carry out linear flow, of course, for plants, linear flow or cyclical flow. And they mutants that are unable to carry out cyclical flow grow well in low light, okay, but not in intense light. And this kind of shows that cyclic flow may be photoprotective, protecting cells from light-induced damage. Okay, you don't want some plants are quite sensitive to light. Okay, so they're very particular on the intensity of light in which they can produce ATP and and undergo an electron flow. Okay. So chloroplasts and mitochondria generate ATP by the same mechanism. Ultimately, we know this already as chemiosmosis. Okay, this is very, very important when you want to compare. Let me just highlight this, put a little star on this. Comparing and contrasting the electron transport chain and the overall procedures between mitochondria and chloroplasts could be potential questions that could be on tests, right? So in both chloroplasts and mitochondria, the ETC, the electron transport chain, pumps protons across a membrane. So what happens here is that this transforms redox energy, okay? Remember, one gets oxidized, one gets reduced as it's going down and down this electron transport chain, ultimately to create a proton motive force, the hydrogen plus gradient. As we know already, similar to mitochondria, ATP synthase will then harness that energy, okay, of the proton motive force to generate ATP, and hydrogen plus ions would diffuse back into that particular area. So just to note here of the locations. So in terms of chloroplast structure, hydrogen plus ions will be pumped, okay, to the thylakoid space, so inside the thylakoid space, and then released into the stroma. Okay. Chloroplasts, they'll be pumped into the innermost membranous space that you can find in chloroplast, and then pumped out into the stroma. For the mitochondria, you have the hydrogen plus ions that are in the matrix. Okay, they're being pumped into the intermembrane space. Okay, intermembrane space between the outer membrane and the inner membrane. Okay, so you're going to the outside. You're creating that gradient in the outside. And in this case, the chloroplast, you're creating the gradient in the innermost side of the thylakoid space. Okay, so just to keep that in mind there. And of course, we'll go through this in the notes. Okay, some electron carriers, including cytochromes, are similar in chloroplast to mitochondria. Yes, they still can fulfill their functions, even though we're in different types of organisms. So ATP synthase complexes are also very similar. It's quite interesting because even though we're evolutionary, different, we're just different organisms, there's some consistencies between the idea of being alive and just evolution, right? So there are some key differences. I've listed here. This is important to know. So in mitochondria, high energy electrons, where do they come from? They come from the organic molecules, right? If you remember glycolysis and the breakdown of to obtain the NADH. Okay, in chloroplast, the electrons ultimately come from water. Remember, the photosystem 2 that is shuttling the electrons from water, okay, passing it down. Okay, so ultimately the electrons come from water. For mitochondria, from NADH, okay, shuttling to here, NADH. And then you have mitochondria using chemiosmosis to transfer that chemical energy ultimately from food to ATP. Chloroplasts use light to turn into ATP. Okay. Mitochondria, we use food to produce ATP. Chloroplasts use light energy to produce ATP. Okay. Spatial organization also is different. So this is what I was talking about in terms of the space. The mitochondrial inner membrane, okay, will pump hydrogen plus ions from the matrix into the intermembrane. So you're going to the outside. Okay, so you're going to get that gradient on the outside of the mitochondria. And then for chloroplasts, the thylakoid, sorry, let me just give it a little space here. The thylakoid membrane would pump hydrogen plus ions from the stroma, from the outside, into the thylakoids. So make sure you understand that particular spatial organization. So again, in mitochondria, hydrogen plus ions diffuse from the intermembrane space through ATP synthase to the matrix to produce ATP. Okay. In chloroplasts, let me make a little. There you go. Because this still talks about number 3 there. All right. So in chloroplasts, you have hydrogen plus ions diffusing from the thylakoid space to the stroma. Again, through ATP synthase. And the catalytic knobs are on the stroma side. Okay. Catalytic knobs are on the stroma side. Just to kind of give you that particular thing here. Okay. The catalytic knob in this particular thing is in the outside. So they're sticking out into the stroma. But in the mitochondria, they're sticking inside within the inner membrane, the matrix of the mitochondria. Okay. Good. Okay. So I'm just going to show this over here just for you guys to have the diagram. This is an overall representation of photosystem 2 and 1 as they're going along to produce ATP and NADPH, okay? Ultimately going to the Calvin cycle, okay? So when illuminated, the thylakoid space pH, referring to pH, drops approximately to 5. The stroma pH rises to around 8. So a 1,000-fold difference in hydrogen plus ion concentration. Okay. So there's a difference because of that proton gradient. So in terms of summary of light reactions, this is something I think is critical. You should know. Okay. Electron flow pushes electrons from water. Okay. Of course, they have low potential energy to NADPH, high potential energy. The light-driven electron current also generates ATP, as we saw, going from one photosystem to another. And then the thylakoid membrane converts light energy to chemical energy stored in ATP and NADPH. Okay. Oxygen is a byproduct. It is not used in any other process, really, in photosynthesis. Okay. Let me zoom in so you can get a nice overall picture here. And let me zoom out. Okay, and then we're going to talk about, for this first part of photosynthesis chapter, the Calvin cycle. Okay, so this is section 10.4. The Calvin cycle uses the chemical energy of ATP and NADPH to reduce carbon dioxide, ultimately to sugar. Okay. So you have this particular diagram here. And depending if you're in the university or if you're studying for the AP program, you might be required by your instructor to study each individual intermediate molecule. That depends. So you make sure you ask on that. Okay, I won't be mentioning these. I will be mentioning these though. So the Calvin cycle regenerates its starting material after molecules enter and leave, similar to the Krebs cycle, right, where you're producing oxaloacetate. Okay. So again, just to note, this is an anabolic process. So from carbon dioxide, a very simple molecule, stable molecule, CO2, you're producing, you're building sugar, and you're actually creating something called G3P, as we know already, glyceraldehyde 3-phosphate. And then these two molecules of these will produce sugar. Carbon, so you build sugar from smaller molecules. Carbon enters via carbon dioxide and leaves as sugar. That's the key point of the Calvin cycle. You do require some energy and, of course, electrons to reduce these molecules to ultimately extract this G3P. Okay. The actual sugar product. So make sure you have this in mind. Don't say Calvin cycle produces sugar. No, it actually produces a 3-carbon sugar called glyceraldehyde 3-phosphate. Okay. And each turn, as you get carbon dioxide coming in, okay, it will undergo this particular cycle. All right. It will fix each carbon of carbon dioxide to produce this organic compound. Okay, so each turn of the carbon each turn of the Calvin cycle, sorry about that, will fix 1 carbon. Okay, so for a net synthesis of a 3-carbon sugar, okay, like this G3P, okay, the cycle needs to go 3 times. All right. The cycle needs to occur 3 times. So it will fix 3 molecules of carbon dioxide. Okay. So to make 1 glucose molecule, you would require the cycle to occur 6 times. So you can fix the 6 carbons of 6 carbon dioxide molecules. Okay. And the general process occurs in 3 phases. So you have phase 1 is called carbon fixation, phase 2 is reduction, and then phase 3 is regeneration of the first carbon dioxide acceptor, which is called ribulose biphosphate. And I'm going to zoom into this as we go along, okay? Or other known as RUBP. Okay, so just for you to see the notes here, not anything extraordinarily difficult there. So what happens in phase 1, okay, carbon fixation. So to fix carbon, it means you obtain that particular carbon and you're going to create a molecule with it. Each carbon dioxide molecule will be attached to RUBP, okay? And this RUBP will come in and get this carbon dioxide molecule, and they will be fixed together, okay, with an enzyme called Rubisco, a very, very, very famous enzyme. Make note of it. You should memorize it, okay? RUBP is also important, so make sure you remember this molecule also. It's the first one that is being used with carbon dioxide as we begin this cycle. Okay, so RUBP comes in. And in this particular diagram, we have 3 carbon dioxide molecules coming in at once. But remember, this happening one at a time, okay? But for simplicity's sake, you have this diagram here saying 3. So 3 carbon dioxides come in with 3 molecules of RUBP, 1 for each, okay? 1 RUBP for 1 carbon. And then as they join together, they will create a very short-lived intermediate. Okay, as you see here, you have 6 carbon molecules. You have 3 of them. Okay, and then this will break down into a 3-carbon molecule. 1, 2, 3. All right, since it's a 3-carbon molecule, imagine you kind of break this in half. Instead of having 3 of these long molecules, now you have 6 of these short molecules, these small molecules. And this is called 3-phosphoglycerate. Okay? And this 3-phosphoglycerate will consume ATP, all right, provide a phosphate group to produce what is called 1,3-biphosphoglycerate. So you've reduced this particular molecule as you come in with NADPH. So you phosphorylated the molecule, sorry, and now you're going to reduce it with NADPH. Okay, so if you notice, there's 6 molecules and there's 6 NADPHs coming in, one for one. And as you're reducing, you're adding that electron, you're removing an inorganic phosphate, a phosphate group is released, and then you have 6 molecules of G3P. Okay. Amongst these these 6 molecules that you have, these 3 carbon molecules, so you have 6 of them, okay? 6 G3P. Only one will leave. Only one leaves the actual cycle, and then the rest, the 5 remaining, will come back to restore RUBP. Okay? So this is entering phase 3. What we just saw right now is essentially NADPH reducing this 1,3-biphosphoglycerate. So this is called reduction phase, phase 2. Okay? So this is probably one of the most important things to know. Amongst these 6 molecules of G3P, only one leaves. And the remaining 5 will be used to re-establish RUBP. Okay? So if you notice, here you have 3 carbons. 3 times 5 is 15. Okay? Through the rearrangement of these molecules and the use of ATP, okay, you will change this whole aggregate of carbons into 3 molecules of RUBP. Okay? Phosphorylating the molecules, you will gain back RUBP to restart the cycle. Okay? So, for every 3 carbon dioxides coming in, you can produce 1 G3P molecule. Okay? Now, again, this needs to happen a total. Remember, this is going in one by one. So, you need to do this 6 times to get a sugar molecule. Ultimately, to get 2 molecules of G3P. Okay? Now, don't get confused. Remember that in this particular diagram, you're having a set of 3 carbon dioxides coming in to produce 1 G3P. So in this particular diagram, okay, you might have different diagrams in whatever test or whatever your instructor is mentioning, but in this particular diagram, you need the cycle to go twice to get 2 molecules of G3P, ultimately to produce glucose. So be mindful of that. It's important to see the number of carbons that you're playing with, right? That you're referring to. Okay. So just to go through the notes here, now that you kind of saw the diagram, okay. Again, carbon fixation, right, is catalyzed by RUBP carboxylase oxygenase. Depending if you're in the university or AP, you might or may not need to study to learn the actual enzyme name, but it's commonly called rubisco, so you can't go wrong with that. Okay, rubisco is the most abundant protein in chloroplast and probably the most abundant protein on Earth. Okay, 6 carbon intermediate is very unstable, like I mentioned before, and ultimately it breaks into 3 carbon molecules. Okay, then you go into the reduction phase where each of these phosphoglycerate receives another phosphate group to produce 1,3-biphosphoglycerate. Remember that one too, because we're going to mention this later on in the later sections. Okay, and then you have a pair of electrons from NADPH will come in and reduce this molecule to produce G3P. Okay. The electrons reduce the carboxyl groups, more specifically to the aldehyde groups of G3P, which obtains a lot more potential energy. Okay. For every 3 carbon dioxide molecules that enter, okay, you get 6 G3P. But remember, you only get 1 of these are actually used to produce a sugar. Okay, so you only get a net gain of 1. Okay, this molecule ultimately, so going into a little bit more information there, this molecule will exit the cycle and be used for whatever you need it to use. Of course, producing sugar in this particular instance. Okay. The carbon skeletons of the remaining 5 will be used to regenerate RUBP. Okay. Again, you do consume ATP for that restoration there. Ultimately, for the net synthesis of 1G3P, and this is important to note, you consume 9 ATPs and 6 NADPHs. Okay? Which is, of course, they are regenerated, they are restored in the light reactions. Okay? Now that you have G3P, it is such a good molecule to, undergo different pathways. You can use it as a building block for different pathways. But it helps glucose synthesis and other carbohydrate synthesis mainly. Okay? So, of course, definitely light reactions and light independent reactions cannot produce sugar on their own. They work together to produce sugar. And because of that, photosynthesis is an emergent property where you're getting the intact chloroplast and that whole complex of molecules and proteins embedded within the thylakoid membranes working together to, execute this process. So it's another example of where you can see emergent properties. Okay, if you just randomly mix them in a solution, it's not going to happen. Okay. So it's very, very intricate how they work together. Okay. With that, we finish the first part of chapter 10. I'll continue on talking about alternative mechanisms of carbon fixation and just a summary of photosynthesis in itself, which I am almost about to finish. Okay. So thank you very much for watching. I hope this video was helpful, and I'll see you in the next one. Take care.